Thank you for joining my talk on social foraging in vampire bats. After almost 4 decades of vampire bat research, we know that these animals maintain social relationships that include social grooming and reciprocal food sharing. We have learned how such food sharing relationships form. There's actually another talk by Jerry Carter on this topic if you want to hear more. And we also learned that these social bonds are even maintained across different social and physical environments. However, this great body of work only focuses on social behavior inside the roost. We still know very little about the role of social relationships outside the roost. Yet social foraging might be beneficial. Saving time by localizing prey or opening wounds, which might take up to 20 minutes, could, for example, reduce predation risk. What we know about social interactions while foraging is based on few anecdotal observations. In the 70s and 80s, researchers reported that female vampire bats often leave the roost in groups of 2 to 6. Several bats may jointly arrive at a cattle pasture, and up to 4 individuals were observed feeding from different wounds but on the same cow. Even wound sharing has been reported, but mainly in mother-pup diets. Aggressive behaviors were reported as well. Vampire bats defend and fight over wounds. Taken together, these observations show that some individuals tolerate each other near food, while others do not. In consequence, Jerry Wilkinson back then postulated that cooperation between roost mates may occur on prey at night in addition to within roosts during the day. However, to test this hypothesis, we would have to closely track the social relationships of vampire bats both inside and outside the roost. That had so far not been possible due to technological limitations. Now we move to the 21st century, and what I am holding up here is the solution to this problem, a miniaturized proximity sensor that I developed as part of an interdisciplinary research group along with electrical engineers and computer scientists. These sensors communicate among each other whenever they come closer than 5 to 10 meters from each other. We can put them on up to 60 bats simultaneously and track all dyadic associations independent of the whereabouts of the animals and at a time resolution of seconds. So we always know who is near whom. Whenever a bat comes near a base station that we typically place inside a roost, it remotely downloads all collected data. In addition, we can use the communication to the base station to determine whether a bat is currently inside the roost or outside, for example, on a foraging trip. We have used this novel technology to study 50 bats from a colony in western Panama that had about 200 bats total. We tracked all dyadic associations among the tagged bats for 8 days and nights. The associations are labeled with a signal intensity value that serves as an indicator of proximity between pairs of bats. We use these association times to create weighted networks for all bats that roosted during day within a distance of up to 50 centimeters from each other. Those were more than 100,000 events. We know from former studies that roosting proximity is a decent predictor of cooperative relationships. We also detected more than 600 dyadic encounters during almost 400 individual foraging bouts. We use these data to answer 3 basic questions. We first ask, when the bats are outside the roost, will specific pairs associate repeatedly across nights more often than expected by chance? The answer is yes. The observed reassociation rates were significantly higher than what we expected from a null model that based on 5,000 permutations of the data. However, only 1 pair of bats reassociated every night, while most pairs only reassociated during less than half of the nights. That means they do not forage in stable groups. In the next step, we ask whether those repeated associations could be predicted by diurnal association rates inside the roost. Again, we found that co-roosting durations predicted both association time outside the roost and the number of nights with repeated associations. This result suggests that social relationships that we usually observe within the roost seem to extend beyond the roost. Finally, we asked whether co-roosting bats would jointly commute to a foraging site. The answer is no. Almost all sampled bats lived several minutes apart. So you might wonder, what causes these non-random associations? Well, we don't know yet. One interesting aspect is communication at the prey. We took simultaneous audio and video recordings near free-ranging cattle and found plenty of social calls during affiliative and aggressive behavior. We even identified a new call type that only seems to be produced while foraging. So in a nutshell, we show that vampire bats do not forage in stable groups. Our results provide evidence that social foraging in vampire bats is not a byproduct of mere attraction to successful foragers, but social relationships extend beyond the roost. The mechanisms that enable reassociation of roost mates at the prey remain to be resolved, and social communication might be one promising aspect. Thank you.